Good morning, everyone. Thanks to Gloria, got our AV technicalities worked out here. I would like to welcome you to this morning's presentation. I'm Jane McPherson. I'm a member of the HAS Science, Medicine, and Technology Committee, and we're happy to bring you today's session. Please take out your cell phones, turn them off, turn them down, silence them, whatever works for you. I had to find mine this morning. So again, if you have a large purse, dig for it now. And then we want to welcome the crowd that's on Zoom this morning. Gloria Goodwin is our course technician who's supporting the hybrid modality today. And as regards questions, Mac doesn't mind you interrupting him, but be sure to raise your hand so we can run over to you with the microphone so everybody can hear the question. And chat people can message Gloria too. So anyway, I'm happy to present McLane Watson, goes by Mac. He obtained his BS in biology from Hope College and then earned his PhD in immunology from the University of Pittsburgh. And then he returned to the Grand Rapids area and is a postdoc fellow at the Russell Jones Lab at the Van Andel Institute in Grand Rapids, and been there since 2022.
Yep.
Okay. And he moved here from Boulder, Colorado, having gone to Blue Lake Fine Arts camp, and fell in love with the area, and it led him to Hope and to our good fortune to have him here today. So, and anybody that's interested, he still plays the cello. So I don't know if we want to invite him for a cello recital or something. Do we have a committee for that? So anyway, I'm happy to have him do part 2 of this presentation. And my apologies that you didn't get part 1 last week, but Ian's going to send both slide sets out when he sends out the evaluation today. So for all of you who had wanted to review before today's course, sorry.
But I'll try and give some more background.
Okay, that sounds good. Thanks.
All right. Thank you all for coming once again. So last week we talked a lot about the immune system and the players of the immune system, so that hopefully we could have a common vocabulary to talk about cancer immunotherapy today. I'll still go over some stuff and have it be a reminder, and hopefully this is a good high-level view of kind of what's out there for cancer immunotherapy in terms of like what's actually being used in the clinic, and then also some things that are being tested and kind of more experimental at this point, but might make it to the clinic soon. So let's get started. Just as an outline, I want to go over the 3 E's of immunoediting, which is really why there's still cancer when we have an immune system. That was a really great question last time of if we have this immune system and it can recognize cancer, why does it still develop? And then I've kind of structured the talk in looking at barriers to the immune response, and then how different immunotherapies address those specific barriers. So barriers to, here, let me turn on this power. Here we go. So barriers to detection, what happens when T cells encounter stop signals, the metabolic environment of the tumor, suppressive cell types, and then physical barriers. And then if there's time, I kind of have a lot of stuff, but we can also talk about my own research. I'll sprinkle in a little bit of stuff that's relevant to this talk as we get to it in these different barriers. So I showed this slide last time, and there's quite a lot going on. But if you remember, cancer develops in this kind of stepwise progression where you have this initiation event that causes some mutation in a cell, but that mutation in that cell is not enough for it to develop into cancer. You need something to push that cell to start to divide. And usually that mutation is in something that regulates cell division, right? And so then when you get it pushed to divide, then all of a sudden it gets out of control because it doesn't have those regulatory mechanisms. So it starts to divide rapidly, uncontrollably, and that's when you end up with cancer. And your immune system is actually quite good at recognizing cancer cells at a very early stage. And that's kind of what this elimination is depicting, is that these cancer cells that arise, they're foreign looking to the immune system. And so your immune system can recognize them, control them, and eliminate the tumors, and you have no problem. And as a normal person, you would never recognize that you ever had this kind of mutated cell that could potentially become cancerous, right? But because of these immense pressures, selecting for cells that essentially can escape this immune destruction, you might get these orange cells that pop out that all of a sudden are a little bit harder to kill from the immune system. Maybe they aren't as recognizable to the immune system, and so they can start to grow out, and you reach this equilibrium stage where the immune system is kind of destroying the cancer as fast as it's escaping the cancer. And so you're in this middle stage. Again, you wouldn't recognize that you had cancer at this point because everything is still technically under control. Nothing's gone crazy. But again, these pressures, you can develop more mutations in these cells that render them even harder to kill by the immune system, or they're more invisible to the immune system. And eventually you can get this escape where these become the dominant cell population, and all of a sudden your immune system really can't control that outgrowth. And that's when it becomes a tumor, and that's when you would notice that, okay, yes, now it's cancer. What's interesting is that these orange cells, of course, are much harder to kill than these blue cells in the beginning. So there's kind of this battle of back and forth between cancer kind of trying to escape the immune system and the immune system trying to kill cancer. So it's like this arms race, and eventually if you get cancer, essentially the cancer has won to some extent. So kind of just to summarize that, the presence of cancer really indicates that the immune system has been overcome. It's actually very good at controlling cancer for most of your life. And then if you develop cancer, that's really when it's overcome. And then the selective pressure of the immune system enriches for cancer cells that survive immune attack. And then cancer itself can produce and support an environment that promotes its own growth. And we'll talk about those these are the barriers to cancer immunotherapy. All right. And so cancer immunotherapy is really trying to overcome those things that cancer is trying to make the immune system blind, or put up physical barriers, or not be detectable, things like that. Okay. I wanted to put this slide up again because I kind of ended with this last time, but we have these various barriers to successful immune response against cancer. So one thing cancer cells can do is, I've been using this term invisible, and we'll talk about this a little bit more. But essentially, cancer cells, we recognize cancer and recognize self through these molecules called MHC, and cancer can downregulate those MHC, so cut them off from their surface so that literally they kind of hide themselves from the immune system. If you don't have those MHC, you can't be seen by the immune system, and cancer will do that. We'll talk a little bit more about that. They can produce these stop signals. That's kind of what's shown here. This is where the most success in cancer immunotherapy has come from. And those treatments are called checkpoint blockade, and we'll go into that. This is really more overview. And then cancer can also consume lots of vital nutrients like oxygen, sugar, and amino acids, which then kind of starve the T cells that are trying to kill and infiltrate that tumor. And then as well as starving, they can produce toxic byproducts as they consume those things. And so that kind of shifts the balance in favor of cancer. And then they can recruit and support immunosuppressive cells, which actually kind of goes along with some of this depleting essential nutrients and producing toxic ones. Oh, and then, okay, so I thought I had one more, but I think that's it. So there are various immunotherapies that try and address each one of these. And they have varying success, and people are also trying to explore more modalities to even try and combine some of these. So it's not so simple as, oh, it's just that the cancer can't be seen, so we just need to increase detection. Because when you increase detection, you might run into one of these other problems where maybe the cells can get to the cancer, but then they see these stop signals, or they get into this really kind of metabolic desert where then they run out of fuel and they can't work. So I think cancer immunotherapy going forward is going to really be a combination of a lot of these different things. But it's a good basis that we've kind of started with these kind of single treatments, see what they do, and then see if we can combine them to enhance the response. So again, this is kind of more of an overview, and maybe I'll just go into it. So I'm going to really be talking about these sort of barriers. So barriers to detection, barriers to stop signals, barriers, the metabolic environment as a barrier, suppressive cell types, and then physical barriers. And then here I just have listed kind of different immunotherapies that are trying to address these. So let's kind of get right into it. So I like to start with detection because this is kind of the first, this is actually the first thing that I thought of when I was going to grad school. I was like, oh, maybe the immune system really just has a hard time seeing cancer, and that's a big problem in the field. And to an extent, it can be, but as you'll see, it's maybe a little bit more complicated than that. So what's really interesting is I have this big kind of confusing graph here. But what it shows is that there are all these different types of cancer. So each dot is a different type of cancer. So like here, this is non-small cell lung cancer, melanoma or skin cancer, colorectal cancer. And then, down here you have pancreatic cancer. And what this is actually depicting is that as you go up on this x-axis here, you have more mutational burden. So those tumors have more mutations per tumor. And then on the y-axis here is response to immunotherapy. And you can kind of see that there's this relationship that as you increase the number of mutations in a tumor, you actually get better response to immunotherapy. And so this is kind of why are some cancers more visible to the immune system than others? Well, the more mutations you have, the more different than self those cancers look. And your immune system is really good at recognizing non-self. Let's see. Here's liver, hepatocellular. And then kidney would be, renal cell. Yeah, right there. Yeah, they're about the same. But you can see like melanoma is one of the highest. And this kind of makes sense because, again, skin cancer arises a lot from UV damage. Like if you go out tanning a ton, that can damage and mutate things in your cells. And usually UV light is a big mutagen, so it can cause lots of mutations, but that makes it very recognizable to the immune system. Whereas pancreatic cancer, this is kind of one of the hardest cancers to treat, and it doesn't really have a big immune response because it really has very few mutations. So essentially it looks like normal self. And then you have stuff that's kind of in the middle. So this kind of gets to how does the immune system determine self from non-self? So as I mentioned, we have these things called MHC molecules, which stand for major histocompatibility complexes. And you have essentially two flavors of them. You have MHC class 1 and 2. You don't need to worry about the specifics of what each one necessarily does, but they do in general the same thing. So these are expressed on every single cell in our body and on the surface of every single cell in our body. And this is the way that the immune system can kind of test and see one is that cell from us, and two, these are very specialized in that they have this peptide binding cleft, or essentially they can hold bits of protein in this little trough and present it to the immune system. So these kind of present what's going on the inside of the cell, gives it a window to the immune system of like, hey, am I infected with a virus or is everything good? And so you kind of are constantly processing and presenting these things, and your immune system is checking and making sure that everything is all good. And this is actually the reason why we probably all heard of like transplant rejection. Like if someone gets a liver and then it's rejected, this is actually the major reason why that it's rejected, because your immune system, we all have these unique kind of combinations. See, they're made up of these like different subunits. We all have these unique MHC molecules that identify us. And so if you put in something that's really different than you, then that's when you could get the transplant rejection, because your immune system all of a sudden realizes, oh, this is actually something completely foreign. This is not me. I need to attack it. Even though, you know, it could save your life if it's like a liver or kidney or whatever. And that's why they try and do a really good job at matching you as closely as possible to a donor, so that you don't get rejection. You want to basically be identical, so that your immune system is essentially tricked into seeing that it's the same. Yeah. Oh yeah, sorry, hold on, one second.
So sometimes the rejection happens years later.
Yes.
Why is that? I mean, we
Yes.
haven't learned to recognize it yet.
No, so that's another problem, is that we have these, this is a major way that the immune system recognizes, and that will stop like acute rejection. So like if you put it in and then like immediately it's gone. But usually over time, those cells are like subtly different enough that your immune system still is able to like kind of recognize it as foreign. And so usually what happens is that over time, you get these like slow little inflammation that then eventually like builds up scar tissue, and then the organ or whatever transplant stops working. So it's that's like minor mismatch is what it's called. And that's really hard to determine. Like no one's going to be 100% match for something else. So usually it's that. It's like these small immune reactions over time that eventually that's why it fails. And 1 second, there's a more for the people online.
Whoever regulates, I'm in line for a transplant. I'm not, by the way, but say I am.
Mhm.
And some doctor is saying, yeah, we're looking for one for you. Who is it who decides whether the match is going to be as close as possible?
So usually there's like these registries, and they'll take like your like a blood sample from you, and then these registries usually have
They make that error.
yeah, blood samples from other people that they have like pre-tested. And so they know it's like, okay, these are the MHC molecules that this person has, and then here's the recipient MHC. And so they can actually go through and search those databases. And then if there isn't a match, you can go and try and find a match, like go and find a relative, because they might have something that's a little bit more close to you. Things like that. So, okay. So, like I was saying, the MHC complexes display proteins to the immune system. That's really their big job. And so if you take this molecule and you kind of are looking top down now, here's what it looks like. It has this little binding cleft and you get these little bits of protein. And so T cells, as they develop in the thymus, are trained essentially on what our own proteins look like. And so they're supposed to be non-reactive towards our self. And if they do react to those self proteins, they actually get eliminated. So they don't get to go out and become mature T cells. So really they get this like training in the thymus so that you don't attack yourself. This is a really good thing. But that means cancer can kind of be a little tricky because it technically starts off as self. And then again, as it mutates, it's easier to recognize because it becomes more like a foreign entity rather than self. So that's why cancers with more mutations are better responders to immunotherapy. So yeah, higher mutational burden gives the immune system essentially more to recognize. So since mutational burden predicts response, how can we boost response to these so-called cold tumors? So in tumor immunology, you have this idea of hot tumor, which is like very immunologically responsive. So there's a lot of immune systems that are a lot of immune cells that infiltrate, whereas your cold tumors are kind of like those pancreatic tumors where there's not a lot of mutations, there's not a lot of detection by the immune system, and maybe you don't have as many immune cells infiltrating. And so there are several things out in the clinic right now that are being used to try and boost that detection of the immune system in especially those cold tumors. So there's these things called oncolytic viruses. People have been working on a long time on cancer vaccines. And then surprisingly, we didn't appreciate, but radiation and then this kind of newer thing called histotrypsy, which we'll go over, is actually works because it stimulates the immune response. So first, I want to talk about oncolytic viruses. So oncolytic viruses are a virus that's actually engineered to selectively infect tumor cells. So how they do that is viruses require certain things to enter the cell and then certain things to replicate itself. And so if they delete one of those genes in the virus that is required for it to replicate itself, highly proliferative cells like tumor cells will essentially supplement that virus with what it's needed to divide. Whereas normal cells, because they're not dividing super rapidly, they will not supplement that virus. So then you gotta get this selective pressure where, again, oncolytic viruses, if they encounter a normal cell, they might be able to still get into a normal cell, but that normal cell doesn't provide the things that it needs to replicate itself. Whereas if it infects a tumor cell, the tumor cell does provide the things that it needs to replicate, and it can go on and replicate and then lyse the tumor, which just essentially means rip it open and then release all those viruses that can then go on to infect other tumor cells. So the idea originally with these oncolytic viruses is that when you treated, really you were just getting destruction of cancer cells by those viruses replicating and then ripping them open. And people didn't necessarily appreciate that the immune system is also involved not only in recognizing the virus, but in the tumor itself as it's ripped open. So if we go back to this diagram, I showed this last week. This is kind of the tumor immune cycle, if you will, where you start here with this release of cancer cell antigens, and that's due to some cancer cell death. So oncolytic viruses are really good at getting in and ripping open tumor cells. And so that not only spreads all the virus, but it spreads bits of the tumor around as well. And these things called dendritic cells are specialized cells in the immune system that will start to eat up those bits of virus and bits of tumor. And then they will take it back to the lymph node and try and find T cells that will react to that. So they'll take all those bits and then go back to the lymph node, present that to T cells, and then those T cells will become activated and go all the way back to the tumor and start killing the tumor. So what's kind of cool about oncolytic viruses is that not only do you get recognition of the virus as this foreign entity, but because you blow up the tumor and you release all these antigens, as they're called, you get response against the tumor as well. So you kind of get these this double response. And that term is called antigen spreading, where you might start off with a very specific, like, okay, where immune system's recognizing just the virus, but as it kind of blows things up, you get more to recognize. So yeah, this is really trying to boost that detection phase and get more things that the immune system can recognize out there. And one of the great things about oncolytic viruses and really all these detection kind of immunotherapies trying to boost immune detection is that they can generate this thing called an abscopal effect. And abscopal just means that there's a response in an untreated tumor site. So for example, this is actually I think it is a PET scan of some patient who's, I think this is the primary tumor that was treated with, in this case, it was radiation, but oncolytic viruses can actually do a very similar thing. But essentially, you can see that all these different black dots are tumor sites. So it's not good. But one site was treated, and then they had this kind of systemic immune response, and you can see that everything's cleared. So this is actually, I think, the bladder. So that is kind of the goal of these immune responses, is to get this abscopal effect, meaning you treat one site and then all the sites in your body that potentially have cancer get eliminated, because now your immune system is alert to, okay, here's what it looks like, here's what is foreign, and then you ramp up. And then, of course, this can also form a memory response. Yeah.
How do you get the virus into the tumor?
Yes, that's a great question. So there's currently, yeah, there we go. So there's currently only one oncolytic virus that's FDA approved and in clinic, and they do an intratumoral injection. So they actually inject it directly into the tumor, which means it has to be accessible to be able to inject into the tumor, right? And so for now, I think this virus is called TVEC, or Immelgic is the brand name. They mainly use it actually for different melanomas and like Merkel cell carcinoma, and so everything's like kind of on the skin and visible. And the goal with this would be to potentially deliver it systemically, like via IV, and then have this response. But I think for now, it works best if you actually give it directly into the tumor. I guess it doesn't have to be the primary tumor. It could, sorry, the question was, does it have to be the primary tumor? I would imagine that would be what doctors go after first. Just because that's also probably the largest tumor pending, but if you have tons of different tumors, then it might not matter. Yeah. So yeah, like I said, there's this one oncolytic virus that's on the market. it can be administered directly into the tumor. And this virus is named TVEC. Monotherapy, so just treating with TVEC alone and nothing else in combination, has about a 26 to 30% overall response in melanoma. So that means the tumors either shrank partially or completely, which is not bad, but compared to checkpoint blockade, which we'll look at next, is it has some ways to go. So people are still exploring how we can improve these different oncolytic viruses. This particular one is a altered herpes virus, which this will not give you the like herpes or any disease like that, but they kind of just use that backbone of that virus and then like, again, genetically alter it so that it does only what the doctors want it to do. So one of the great things about oncolytic viruses is that you could use them to deliver different payloads. And by payloads, I mean drugs or different cytokines, which can boost the immune system, things like that. So they have here, they have like this very big DNA genome that you can insert genes into. And because it's targeted to the tumor and because you can inject it directly into the tumor, essentially as these viruses replicate themselves, they can make whatever product kind of you want. So you can, for example, in my graduate lab, we made these viruses express leptin, which leptin is the thing that helps make you hungry. And for whatever reason, it actually, having leptin in the tumor makes T cells really fit and can kill tumor cells better. And then you can also include these different inhibitors in there. So again, you can kind of use this thing as a drug delivery system, which I think going on with oncolytics will be kind of the next step of where this is going. Yes.
I am just intrigued by how the thought process of a scientist can take a herpes cell and think it can be used to combat cancer. How does that, how do scientists make those kind of connections?
Well, actually, that's a fairly good segue into this next section.
Okay.
I'll
wait.
Yeah. So it's not exact, but I'll hopefully this will explain it. Cancer vaccines have actually been around, or at least the idea of them has been around for quite a long time. So this guy named William B. Cooley in 1891, he's a doctor in New York treating cancer patients. And there was a patient that had an inoperable tumor, and he decided, and I don't know, maybe he was crazy. And that's how you discover things, is you just try stuff. Yeah, is that he saw this inoperable tumor. This person didn't have a lot of chances. And so he actually injected this bacteria into the cancer, thinking maybe if I, inflame this area, that the cells that attack the bacteria will also attack the cancer, and maybe we'll see if we get anything. And that's actually what happened. So he infected this site, and then all of a sudden, this person got better. And it wasn't only this person, but he treated over 40 years, 1,000 different patients with bacteria and bacteria products. And so he's really considered the father of immunotherapy. And so kind of from this, I think, similar idea came oncolytic viruses. So it's not bacteria, but if we can induce something, like use something that the immune system already knows is really, we know that the immune system is very good at attacking viruses and bacteria. If we can use that to our advantage and maybe trick the immune system into also recognizing cancer along the way. So maybe he was kind of a mad scientist and did it, but the thing is it worked. And that was really cool. And they didn't actually know why it worked back at this time. And what's interesting is even though this happened, it kind of dropped off as radiation therapy and chemotherapy became more of a kind of standard of care. And people forgot about kind of what's termed now as Cooley's toxins. And that maybe the immune response could have something to do with cancer. So there was a really long period of time where people thought you were absolutely crazy if you said that the immune system recognized cancer at all, or that could be, a cure. And that was really only changed recently in like kind of 2000s when the first immunotherapies were approved. Yeah.
Sitting in the front row where they. But it looked like it took 15 years before they finally recognized it. We discovered that.
Yeah.
So, yeah,
Definitely. And I'm sure that people probably thought he was a little crazy, but and people with cancer were desperate and they saw some success. So yeah.
I have a question here from online. Is the role of gamma globulin shots for a person born with extremely low white blood cell system?
That I am not entirely sure.
Okay. She does have another question.
Yes.
And is there a correlation to cancer susceptibility and successful eradication?
Yeah. Okay. So for people who are immunocompromised, they usually are at a higher risk for cancer. So that's just another kind of evidence for the immune system being able to recognize cancer. And actually, that was one of the things in the 80s when HIV and AIDS kind of epidemic hit. Those people that developed HIV and AIDS were at a higher risk for this type of sarcoma. And the doctors were kind of confused as to why. But HIV and AIDS, the HIV virus targets your CD4 T cells. And so essentially like wipes out a very important part of your immune system. And then that leaves you more susceptible to developing different types of cancers, as well as getting like fungal infections and like all these other things. So absolutely is immunocompromised cancer. Yes.
I'm interested in when you talked about how in 2000 immunotherapy started kind of maybe being more recognized. So when you talk about chemotherapy, radiation, immunotherapy, where are the dollars going? Where are the successes? And I know it depends on the type of cancer, but where's NIH putting its money and your Cancer Institute?
Yes, definitely. The shift has gone to immunotherapy. And drug companies, especially, as I'll show with these like checkpoint blockade molecules, once they saw that those were having incredible success, almost every drug company started trying to develop their own version of those checkpoint blockade drugs. So there's big investment into that area. So I think people really recognize that the immune system is a huge part now, and that cannot be ignored. And so we really need to shift our focus to immunology and cancer, not just like looking at the cancer itself. Yeah. All right.
In those days and shortly thereafter, it was not uncommon, my understanding, that they would, you'd try to find ways to jack up the immune system by instilling something that the body can treat, in hopes that the jacked up immune system will then treat things that the body that you don't know how to treat.
Right.
And that was done for a variety of things, including syphilis and a bunch of other things.
Oh, interesting.
So I think but this is an interesting link. It's very, very interesting thing.
Yeah. All right. Just so we can hopefully get through most of this. These are great questions, though. I really appreciate this. So in the modern age, we're not necessarily injecting bacteria and bacteria products as vaccines for cancer, but trying to do it a little more targeted. So there are different ways, and this is kind of a complicated diagram, but I'll try and break it down. There are different ways that you can kind of immunize, or people are trying to immunize the body against cancer. And two, there's this idea of a therapeutic vaccine versus a prophylactic vaccine. So a therapeutic vaccine would be you come into the doctor's office, you already have cancer. So you're not trying, like at that point, you're not trying to prevent cancer because you've already got it. And so you want to treat it with a vaccine. So that would be therapeutic, whereas prophylactic would be like, I don't have cancer yet. I don't want to get cancer. So can I essentially immunize myself to prevent cancer coming all together? And so essentially there are various different ways of trying to immunize your body, but all of them are essentially providing those cancer antigens, as they're called, or those pieces of cancer to these specialized cells called dendritic cells that will then stimulate a T cell response to recognize cancer. And so you can do that through in situ, which is essentially like what we talked about with the oncolytic viruses, where you actually just kind of blow up the tumor as it is in the body, and then hope your immune system kind of picks up those pieces and then can stimulate a response. You can also take out tumor cells and grind them up, and then give them back to the body, or put them into these dendritic cells outside the body and then put those into the body. And then, of course, we have in this kind of technological age a way to sequence cancer cells and find out, here are all the different mutations that are occurring in this cell. Can we essentially find those mutations, make those proteins, and then give them back to the person to be immunized? And this is kind of like a shared, or this is a personalized medicine way. And this is obviously way more labor intensive because and changes from person to person, because depending on the mutations of one person's cancer versus another person's cancer, this can be very targeted to an individual. But then you can also kind of do a similar thing where lung cancers, for example, have a very common mutation. Like most of them have this common mutation in this gene called KRAS. So you could essentially make a vaccine that is against this mutant KRAS. Yeah, but either way, all of them are trying to provide your body with those bits of cancer to then be able to recognize and create a T cell response against it. So what vaccines are actually currently approved? These are the preventative or the prophylactic ones, and you've probably heard of these. Like Cervix is against HPV, which HPV can cause different types of cervical cancers, or most cervical cancers. And then Gardasil in a similar fashion, it can protect against cervical cancer, head and neck cancer. Gardasil 9, which just extends what HPV viruses that it immunizes against. Again, is like genital warts, cervical, head and neck cancer. And then hepatitis B, which can prevent hepatitis B related liver cancer. So these, you can see that there are some cancers that are actually virally driven. And so that's nice in the development of vaccines, because we know how to make a vaccine against a virus. And so if you can just prevent the virus from infecting, you can prevent that virus from pushing cells to become cancerous. What's harder is the therapeutic vaccines. Here, this particular BCG is actually more like Cooley's toxins, where it is using a weakened bacteria to stimulate the immune system. And this is actually approved for patients with early stage bladder cancer. And the only kind kind of like true cancer vaccine that's approved right now is it's called Provenge, and this is actually used to treat prostate cancer. So this is where you take your the patient's own those specialized dendritic cells, and then give them bits of the cancer and then reinfuse them back in the patient in hopes that then they can recognize the prostate cancer. Overall, though, cancer vaccines have had really poor success. And people have been working on these and trying, and there's just not been a lot of movement in terms of cancer vaccines. But this also happened with things like checkpoint blockade and CAR T cells, and things that are really successful now is they had this kind of slog through the research of it just not working until they all of a sudden had a breakthrough. And then, so people are trying.
Who gets the prophylactic vaccines?
These can be, I mean, a lot of young women are recommended to get like Gardasil, even boys too, just to prevent HPV infection. So I don't know, it's kind of out in the general public now. And it's up to, it's not like a required thing to like go to school or anything, but anyone is able to get it. Yeah. Yeah,
Why wouldn't that be a common vitamin or pill-like thing that everyone takes starting at age 15?
it is now recommended. These are actually right. Yeah, so this is the question is like, why aren't we all taking these vaccines? And now, so the first off, these are actually relatively new vaccines, as in like I think post 2000. And originally they were like just recommending it for young women, but then they realized what you did, like it's going to be good for everyone if you can just prevent HPV infections. So now they're recommending it for like, you know, the young teenagers. Yes, but it can also, there are different types of HPV. So some HPV is sexually transmitted, and some like what cause head and neck cancer are not necessarily sexually transmitted. So there's different types of HPV. Yeah.
Is the question about the Hep B vaccine, why is it required for incoming college students and not others?
It's required for healthcare workers. It's recommended for a lot of adults these days, just because it's preventative. And if you don't get liver cancer or chronic hepatitis from hepatitis B, it's a boon, you know, and hepatitis B can be transmitted in various sexual ways, back to that, and also IV drug abuse and other mechanisms for people who use blood products like hemophilia. So it's just become a more common thing, and that's why.
Yep. Yeah,
Okay.
exactly.
Sorry.
No, that was perfect.
Also, just curious, have you seen or heard of any application that where AI might be of benefit as far as testing, development?
Yes.
Anybody looking at that?
Yes, I am sure that they are. I don't know specifics, but I do know. So this has actually been a big thing in the structural biology field, is that I can't remember what the program is called. It might be like AlphaFold or something, but they have used AI to better predict when you give it a protein sequence, how that protein will actually fold, which is super important for developing vaccines, because it's all about the immune system recognizing a specific shape of a protein. And so I'm sure people are using AI to predict that, to see what possible mutations would look like, what possible mutations are out there. So 100% people are diving into that, yeah. Okay. So this is something I feel like I was surprised to learn about in my schooling, is that radiation is actually a form of immunotherapy. So we've all probably heard of radiation treatment for cancer. This is really standard of care, and it has been a pillar of cancer treatment for a really long time. And of course, this is where you would take a beam of concentrated radiation, and we've gotten better. We used to just do whole body radiation, which is obviously not very specific and could damage non-cancer tissues, but now we can really hone in on the tumor and shoot it with this radiation and essentially just blow up tumor cells, right? It heats it up and then rips them apart. But recently, as I said, it's been appreciated as immune accelerant because you get this ripping of the tumor cells apart, and you get these antigens that get flung everywhere, and your immune system can then pick them up and then present them to the T cells. This is actually really an immune response. And so much like vaccines or those oncolytic viruses or the other things we've talked about, you're trying to inflame the tumor to get the immune system to then recognize and attack. And then the hope is to induce that systemic abscopal effect where you can treat one tumor, and then even if there are tumors that aren't visible or reachable, that your immune system can go and get. And then something that I actually learned about very recently, this was maybe like a month ago, a researcher from the University of Michigan came and gave a talk about this thing called histotrypsy. And this is also a form of immunotherapy. So histotrypsy actually uses these really high frequency ultrasound waves. So think of it like a ultrasound machine on steroids. And you can use it very specifically. So you can pinpoint where you want to put those high ultrasound waves. And like radiation, they can lyse tumors. So, sound waves are essentially just vibrations. And what happens is when you aim it at the tumor, it vibrates and then creates these what are called cavitation bubbles. And so there are these holes that these bubbles that expand and then collapse really, really quickly. And that ends up ripping up the tissue and releasing antigens. However, what's super exciting about this is that unlike radiation, it doesn't produce a lot of heat. And heat, in terms of the immune system, can actually be bad because when you heat up proteins, they don't keep their shape. They unfold and they fall apart. And so then they are, not quite as recognizable in their kind of native form. And so what this can do is just with sound waves and very, very, very minimal heat, blows up the tumor cell, releases all those antigens, but keeps those antigens all intact. And what's cool is that this researcher showed that it's able to induce an even stronger response than radiation and some of these other techniques that have been used to induce this immune response. And I think because it's that lack of heat, that you're actually able to get better recognition. So this is super exciting also because it's non-invasive, because ultrasound can reach, it can penetrate through bone, it can essentially reach any part of your body. And again, you don't have to make any sort of incisions or anything like that. And so I'm excited to see where this goes. It's not necessarily widely used now, but again, University of Michigan is investing into this, and it's creating these really strong immune responses, and people are trying to combine this with other immunotherapies. And I just like the fact that it's kind of this simple thing that we've always had is ultrasound amped up, and then you can reach anywhere in the body, non-invasive, and it just has this huge response. And you heal well from it too. Radiation sometimes creates a lot of scars, I think, again, because of the heat, like internally, but for whatever reason, this disruption of these cavitation bubbles keeps everything fairly intact and doesn't create scar tissue. So I think this is really exciting. They are. Yeah, they're trialing it on patients. Yeah, the question was, are they using it on patients? Sorry for the people online. Yes, so they are using it on patients, but I think it's just in trials right now. And then like they have preclinical models and like mouse models and things where they've combined it with other forms of immunotherapy, and it's amazing. It can like clear tumors. It's pretty cool. No, so that's the other cool thing, is that it doesn't have to be a specific cancer type, which is like a lot of these immunotherapies, they actually work for multiple cancer types. Which is a good point, because the previous cancer therapy, like chemotherapy and other things, people developed drugs for melanoma or a specific type of melanoma or specific type of colorectal cancer, because they looked at those mutations in those cancers and said, okay, we're going to block this pathway, which is really important. And so it only worked for one type of cancer, but immunotherapy, because it's not necessarily specific for those things, it's just cancer in general, it can work for different types. Yeah.
So the idea here is is just to and create a tremendous stimulus or a tremendous recognition ball for the immune system, because those antigens will still be much less attractive to the immune system if they're more like self, like a pancreatic cancer cell,
Right.
versus a melanoma, which is very unlike self because it's
Right.
high. So, but you're bridging that difference by just having so much antigen out there. Is that the idea?
Yes, and when you have tissue damage occur, which is this is a good point. Your immune system has a lot of regulatory mechanisms in place for good reason, because you don't want to just create responses against yourself when they're inappropriate. And so context matters. When you start to blow up these tumor cells, you get like tissue damage, and tissue damage, along with those antigens released, you get these things called danger associated molecular patterns, or DAMPs. And those create the context of when the immune system sees all those antigens and sees those signals that, oh, this is an emergency, like this is actually inflammation, then it will react. If it doesn't have those kind of DAMPs, it's not going to react as much.
Will it also damage the normal cells that have very similar antigenic appearance to the tumor cell and other places in the body? Is that a side effect that you don't want?
Yes, there's a side effect that you don't want. That's not, I mean, usually that's not actually what's seen. So that is always a fear of cancer immunotherapy, is that all of a sudden you're going to ramp up the immune system too much, and then it's going to recognize non-cancerous tissues. For the most part, I don't know exactly with histotripsy, but I think it's pretty localized just to the tumor region. Yeah, thank you. Yeah. The question was, is histotripsy being trialed anywhere else except U of M? And I do believe that it is being trialed other places. I can't say for sure. I just know that there's a big research interest there, because a researcher and a clinician have kind of invested into that and formed their lab around that. So, all right. Yeah. Just in case it doesn't work. Okay. So now I'm going to kind of move on to the stop signals as a barrier to cancer immunotherapy. So kind of like this little cartoon. Here we have cancer cell, it's got a bat. So some T cells recognize and target cancer cells. It's got this picture, little bow. And but before the T cell can actually kill it, the cancer cell might fight back using this thing called PD-L1 to target PD-1 on T cells, and then kind of knock it unconscious and go to sleep, and then have the tumor cell escape. So tumor cells can express these stop signals. So that's what this PD-L1 is. And these stop signals put the brake on T cells trying to attack it. So again, they have no problem getting in and detecting the cell. But once they get there, the cancer hits it over the head with a bat and the T cell goes to sleep. So checkpoint blockade is trying to stop or block these negative signals, essentially put a hard hat on this little T cell so that it can survive being hit with a bat. And how they do that is they use manufactured antibodies. We talked a lot about antibodies last week. These are small proteins that are very specific for different, again, antigens is the technical term, but like bits of cancer cell, bits of virus that can very specifically recognize them, and then they bind all over them. And essentially they can coat stuff, which makes it easier for the immune system to eat, easier for the immune system to kill. And then it also, of course, blocks any interaction if you're surrounded by all these little proteins and you can't actually interact with certain receptors. So that's what these antibodies do. And so the main ones out right now are against this molecule called CTLA-4 and against PD-1. So I'm going to start with anti-CTLA-4. This is actually the first checkpoint blockade immunotherapy that was approved in 2011 by the FDA. And the person who did this discovered this was James Allison, and he's actually at MD Anderson and runs his own lab there. And in 2018, he won the Nobel Prize for medicine for this discovery. It's that big. He also co-won it with the guy who discovered PD-1, which I'll talk about in a second, Tsuko Hanjo. He's in Kyoto University in Japan. So they shared the Nobel Prize that year, but that's how big an impact this was on the field. So like I said earlier, T cells actually need multiple signals to activate to safeguard against accidental activation. You don't want your cells just going and attacking your body at any instance. And so they've created kind of these different regulatory mechanisms that prevent accidental activation, if you will. And so you need several different signals to get activated. So not only does the signal one, this is the T cell receptor, and it recognizes its antigen or that bit of cancer or bacteria or whatever. So that's one signal, but then it usually needs this other signal called signal two. And signal two are these activation receptors. You have this stimulation plus signal two, then the T cell knows that it's okay to activate and it can go. So one of those signals is this molecule called CD28, and it interacts on this antigen presenting cell or this dendritic cell, as I've said before, with this molecule called B7. So you need this interaction plus the T cell receptor and the antigen interaction in order for them to activate. But there is this molecule called CTLA-4, and this CTLA-4 is expressed by the T cells. And what it does is that it recognizes this B7. So now it's in competition with the normal receptor for this ligand, as it's called. So it is actually a stronger binder of this B7 than the CD28 is. And so what ends up happening is that it competes that CD28 away, and then now all of a sudden you don't get that second signal. Essentially, it blocks that second signal, and then the T cell doesn't activate. Let's see. So what the drug is called ipilimumab, and so this is a manufactured antibody. What it can do is it will actually bind to the CTLA-4 and prevent its interaction with this B7, and then allow the normal CD28 B7 signal 2 to occur. And then that means the cells can then activate. So they've got signal 1, they've got signal 2. And then that unleashes the immune system to be able to go and kill the cancer. All right. So in a similar way, anti-PD-1 therapy targets these kind of inhibitory receptors on T cells. So once T cells are actually activated, they circulate through the body and they find their antigen presented by cancer cells. So here's a tumor cell here, here's a T cell here. It recognizes this interaction, and that's good. So CTLA-4 is really involved in activating T cells, and PD-1 is involved after those cells are already activated. So they go to the tumor, they engage with their antigen, and the tumor cell actually expresses this thing called PD-L1, which binds to PD-1 and turns off the T cell. So that was like the one with the bat. It turns off the T cell. So the T cell then says, oh, I don't want to kill this cancer cell. It's sending me the stop signal. And normally, when T cells see their antigen, they actually do turn on PD-1. Again, this is another regulatory mechanism that they just want to make sure they're not accidentally getting activated. And if they are accidentally getting activated, or they get activated too much, then there are these kind of steps to bring it down so that there's not all this collateral damage. So PD-1 in its normal context is supposed to prevent overactivation. However, when you constantly see your antigen, as in the context of the tumor, you're constantly getting this interaction. It's constantly having the T cell express PD-1, and then that really high expression of PD-1 really dampens the T cell, and that's when it's a problem, because all of a sudden now you have more PD-1, so you have more negative signal than you have positive signal to kill. And so that's what I said here. Persistent T cell receptor stimulation causes high PD-1 expression and shuts down the T cell. And so what these antibodies do then is that they bind PD-1, much like they did CTLA-4. Here, it's just shown as this triangle. And then people have also made antibodies against PD-L1, which blocks and binds to the tumor cell here. But either way, it's preventing this interaction from occurring. And then that unleashes the cells to be able to go and kill cancer. And as I had said earlier, checkpoint blockade has seen the greatest success in cancer immunotherapy. It was given the Nobel Prize in 2018. This is a kind of overwhelming diagram, but just showing you the number and types of cancer that PD-1 and CTLA-4 are used to treat. So you've got head and neck cancer, melanoma, Merkel cell, liver cancer, kidney cancer, these different colon cancers. I mean, almost every single part of the body, right? And let's see here. You have really strong responses with a combination too. So people aren't just treating with CTLA-4, anti-CTLA-4 or anti-PD-1. They're usually treating with some combination of both, and that really boosts the immune response, especially in melanoma, where you can get responses of like 70%, 80% of patients are having responses, meaning their tumor shrinks or is completely gone. And that's one of the things that we hadn't really seen before in classic cancer treatment, is that PD-1 and CTLA-4 provided this durable response. So sometimes patients would completely clear their cancers, but then the cancers wouldn't come back. And that really had not been seen with any other prior cancer drugs. So that's why people got very, very excited about this. Everyone's producing a PD-1 antibody, everyone's producing a CTLA-4 antibody, and trying to get it approved. And as we talked about earlier, this correlates, response correlates with higher mutational burden. So higher mutational burden, better response to PD-1, again, because the immune system recognizes more mutation tumors. And then also in the research field, it's a little funny because everyone wants to get their research published and have it be a big thing. And so usually they come up with some sort of novel drug, and then they, just for fun, throw in PD-1 in their treatment and show that it, synergizes with the treatment. So literally everyone's just throwing in PD-1 and CTLA-4 everywhere. And what's great too is that it has minimal side effects compared to chemotherapy. So classic chemotherapy is really just essentially these toxic drugs that are supposed to kill rapidly dividing cells quicker than your other cells, but it's still toxic. And so you're just hoping it kills the cancer before it kills you. Whereas this is very targeted. Again, those antibodies are very specific and recognize very specific things, especially PD-1. That PD-1, PD-L1 interaction is really high in the tumor, but it's not really high everywhere else. So even though you treat systemically, it kind of ends up being very specific to the tumor because PD-1 and PD-L1 are really only expressed on cells in the tumor. Where CTLA-4 has a few more side effects because you're kind of treating generally T cell activation. But again, that actually has, it's been better than chemotherapy for sure. So. And I just want to show this too, that these are single, this is just treating with either anti-PD-1 or anti-PD-L1. And you can see that a variety of different cancers have different response rates. So like in Hodgkin's disease, you have a objective response rate of 87% of people respond to that, which is just absolutely insane. That, even in liver, which is hard to treat, you have 20% of people respond. So it's truly an incredible breakthrough. But that also leaves a big question. Yes, there are 20% of people that respond, but that means 80% of people aren't responding. And we have no idea why. So a lot of researchers are figuring out, how do we boost response to these drugs? What is actually preventing, on top of, this PD-1 and CTLA-4, what's preventing the immune system from doing its job? Yes.
Do they know for sure that the lymphatic cells have this,
I think they do. So sorry, the question was, do they know for sure that the hepatic cells or liver cells express PD-1? And what's interesting is that people have looked at that as a way to try and predict who's going to respond to PD-1, who's not. And it doesn't necessarily correlate with PD-L1 expression, even though that would be the kind of the obvious answer. So yes and no. Like sometimes they do in like some cancers, it's like, yeah, PD-1, PD-L1, you're probably going to respond, but that's not true for everything. So it's also a little confusing. It would be nice if it was really straightforward. Okay. So this is kind of where my research falls in, is this metabolic barrier to cancer immunotherapy. And so I think I mentioned last time that cancer cells are kind of metabolically deranged. So tumor cells themselves consume lots of essential metabolites like oxygen, amino acids, which make up proteins, and glucose. And all of those things are essential for T cells to divide, to kill tumor cells and function, right? So you can imagine when T cells get into that tumor, they're all of a sudden in competition with the tumor for these essential nutrients. But the tumor is actually much bigger and has better importers, and so it usually wins out. And not only that, but the tumor cell as it eats up these essential nutrients, produces these toxic metabolites like lactic acid, kynurenine, or reactive oxygen species, or ROS. And those tend to dampen T cell responses. And so you kind of get this double hit where they're starved, but then they also get these like bad metabolites that they have to deal with. So T cells that make it into the tumor have to fuel themselves in this metabolic desert, and they're in competition with the tumor. And so this is a little bit what I did in my PhD is I actually focused on these cells called regulatory T cells. And so I kind of wanted to use this as an example of how the metabolic environment creates a barrier not only to suppress immune cells in itself, but also support immunosuppressive cells. So these regulatory T cells, or Tregs, are the natural suppressors of the immune system. So of course we need things to go out and attack and clear, but once you've cleared the virus, you've cleared cancer, you need to rein in your immune system so that you don't get this rampant inflammation that's happening all the time. And so that's what these regulatory T cells do, is they bring down the immune response. But cancer takes advantage of these cells and is able to recruit them and support their proliferation and support their suppressive function. And of course, that then creates this immunosuppressive environment within the tumor that prevents those good T cells here from actually killing the tumor. So like I said, tumors take up lots of glucose, also known as sugar. And as a byproduct, they put out this thing called lactic acid. And we've probably all heard of lactic acid because if you work out, you know, muscles get sore. That's actually lactic acid. So tumors are actually really concentrated in that because they're kind of constantly metabolically going, consuming these things, and pumping out tons of lactic acid. And it turns out, I discovered that these regulatory T cells actually like this lactic acid, where they can take up that kind of what's thought of as a toxic or like waste product metabolite, and then use that to support their suppressive function. And that when you actually get rid of the ability of the Treg to use lactic acid, then all of a sudden they are no longer suppressive, and the T cells can then come in and wipe out the tumor. So this is I think a good example of how metabolism can play into being a barrier, is that, you know, not only are these good metabolites consumed, but these toxic ones appear as well. And then that supports different cell populations that might be pro-tumor or pro-immune system. And of course, this is not supposed to be like I'm going to go through this. This is just to show you that this is there's a lot of complex stuff going on, and there are lots of metabolic drugs being tested out on the market. And none of these drugs also, these are all in trials for treating cancer, but they're not necessarily thinking of the immune component. But that's okay because there's a lot of researchers, including myself, who are interested in, okay, what happens if we block the tumor cells from, you know, taking up glucose, or if we block them from releasing a bunch of lactic acid, or kind of name your metabolite or pathway? Can we level the playing field and give T cells a better chance at competing for the things that they need to fuel themselves and then kill the tumor? And so it's really exciting that a lot of these are out on the market, and then people are starting to combine these with like the checkpoint blockade molecules to see if we create a better metabolic environment and then combine it with an enhancement of the immune system, do we get this really, really good response? And the answer is yes in a lot of cases. So this is cool. Yes. The question.
Do they know what to prescribe?
I'll repeat your question then, yeah. So the question was, does a family doctor who you've had for 20 or 30 years know all this and know what to prescribe? And the answer is, depends on how good your doctor is, but probably not a primary care physician. And you would probably be, if you had some sort of cancer, you would be like kind of referred out to an oncologist who they should hopefully be up to date with all the new stuff, right? And what's hard too is that there is like a lot of this stuff, they're in clinical trials, so they're not FDA approved. So doctors everywhere can't just like give out stuff. Like the checkpoint for sure is FDA approved, and that's actually frontline treatment. But so if the system is hard to see if like they have a route that they need to treat you with first, and then if you don't respond, then they go to this other thing. There's kind of this protocol. So Mhm. Yeah. Well, so the question is, they don't really, basically, where can we go to get the kind of care that we need to prevent cancer? And I think that is still an active area of research. People are not, we don't have a definitive answer on do these things, and this is going to be the outcome of like preventing cancer. It's not an absolute yet. And people also like, yes, there's like lots of anecdotes and stories, but researchers, like we don't have a lot of evidence yet for it. That doesn't mean it's not true. It's just people haven't studied it, and people haven't, like the government necessarily hasn't paid for people to study it. So
Teaching it in medical school.
I think so.
Yeah, I mean, yes, I mean, well, I'm obviously out of medical school now, but even watching nursing students, they're learning a lot about these things. And we're not experts. You wouldn't want your family doctor to do your open heart surgery. You really need to see the right specialist.
Yeah.
And the family practice doctor is sort of a generalist with knowledge of lots of things, but not drilled down so that we could recite this whole chart by heart.
Yeah.
You know, that's why Mac's a researcher. And I worked in an oncology clinic for a number of years myself, and it's become very specialized. So you have someone who's just doing melanoma, and your family practice doctor might send you to an oncology clinic specifically to see the melanoma specialist, because it really is drilled down like that
Yeah.
now.
Yeah.
So the day of even general oncology is sort of fading. It's too much.
Bob's question also is, you can help with that a little bit, is I think people know that, you know, from the history of cancers and tumors, what are the likelihoods? What are the warnings? What is a group? Are you in a group that has more likelihood of developing these things? And then you have either chemical detectors or screening tests that help to put you in or out of a risk category. And those aren't perfect, obviously, but but those steer a lot of people toward getting more routine exams to detect things. You know, routine colonoscopies, for instance, on those that have high risk factors, or other familial kind of markers. Some people in some age groups are more likely to get things than others, and so that's what the primary care or any kind of triage group in the medicine whole catalog would do, is put you in a group of higher or lower risk for things and do screening things to hopefully pick things up as early as possible.
Yeah. Thank you. All right. Kind of one of the last main things I want to touch on. We might not get to everything, which is okay. Because I'm really enjoying this discussion, and this is for you all to learn what you want to learn. So this is really good. But I think I do want to touch on CAR T cells because this is another kind of breakthrough therapy that you might hear about a lot in the news as well. So what are CAR T cells? CAR actually stands for chimeric antigen receptor T cells. And what they are is they are engineered T cells that recognize your cancer. So in this scheme, this is how they work. Healthcare providers will actually collect blood and then pull out the T cells from a patient. So now you have these nice isolated T cells, and then you can actually give them a T cell receptor that you know is expressed by the cancer. So you just kind of turn it towards the cancer. And so that's kind of depicted by these yellow spiky things, is that now they can have this receptor that specifically recognizes the cancer, and they know it recognizes the cancer. And what they do is then outside the body, they grow up in these flasks, millions and millions of these T cells, and then reinfuse them back into the patient. So it's this really unique therapy where you can take T cells out of a person, manipulate them, grow them up, and then stick them back in the person. And this has actually worked really, really well in blood-based cancers like leukemias and B-cell lymphomas. So that's just meaning like the cell of origin for that cancer is a blood-based cell. And so it's worked fantastic. Pediatric patients have been treated with these CAR T cells, and it's worked really, really well. But it's not had such great success in solid tumors, which solid tumors essentially is everything else that's not, leukemia or any blood-based cancer. And so people are really trying to work hard at figuring out how can we make those CAR T cells better. And as we've been talking about, all these different kind of barriers that the immune system hits, is that CAR T cells are going to hit all these same barriers. So they might recognize the tumor, but once they get to the tumor, they're going to see, PD-L1, and they're going to get downregulated by CTLA-4, and they're going to enter this kind of metabolic desert. And so what's a lot of focus, and including some of my own current research, is actually looking at, we have this very unique opportunity of growing up the cells outside the body, where we can manipulate those cells, do very specific things to those cells without affecting everything else in a person, right? It's completely separate. So there's not going to be like side effects. And also if we do something wrong, those cells die, but like the person is going to be completely fine. We're not going to put them back in. So a lot of research is figuring out how can we make these cells more persistent? How can they get into the tumors and maybe overcome these metabolic barriers? So one of the ideas is that you can take cells, and the media that you expand them in is not like the blood at all. It has very, very high levels of glucose, these different amino acids, all these different metabolites, and it's missing a lot of metabolites that aren't in the blood. And so that kind of gets them addicted to these certain things that are in excess outside the body. And then once you put them into the body, they're like, where'd all that go? And then they die and they don't do well. So people are looking at adding those things back into the media, making it more like blood, and growing up that way, or blocking certain pathways as they grow them up so that they don't become addicted to certain things that you don't want them to. The other thing that we can do is we can overexpress these kind of metabolic genes. This is something we did in my grad lab where we took T cells and we overexpressed this thing called PGC1 alpha, which essentially just makes more mitochondria. And mitochondria are the powerhouse of the cell. They create energy as well as other intermediates and things that cells need. So if you give them a lot of mitochondria, essentially they're really strong, and then they can persist. And then you can also delete these kind of negative regulators of metabolism. And if you get those out of the way, again, it might unleash the T cells to be able to persist and kill cancer. And then I guess, are we at time?
We are. Sorry.
All right.
I just have a quick question. Does the Van Andel Institute have outreach programs to educate doctors about their work and the results?
That is a good question. I don't think we do on the clinical side.
I can answer that.
Yeah,
They do in terms of reaching out to oncology groups
okay,
and they invite them to tour the Van.
okay. I guess I'm
So-
just not involved.
So, and then they do their spiel. So, yeah, they have outreach, but not to my knowledge, not to lay people in the community unless you're invited out to speak, right?
Right, yeah, they have those, and then I guess, more recently, we do have public lecture series that go on throughout the year, maybe like 4 lecture series. So I am speaking in that at the end of the year, like December. So, yes.
Of the treatment for what we finished with the virus, coronavirus.
Mhm.
Where, if in fact we found, whether it was engineered or not, a virus that needed to be addressed, cancer virus. And we had to look at immediately where we would put in so much resources like we did for fighting the virus in 2020. Where do you think the best bet, where that money would go? Where would we find the most bang for the buck, if you will?
I think I'm biased because I am involved in metabolism, but I think from what I've seen in my own research and the other research in the field is that I think the metabolic angle plus the immune system angle is really strong. We've had, I mean, at University of Pittsburgh started this trial where in preclinical models, we actually treated mice with metformin, which is, a diabetes drug, but it's like a general kind of metabolic inhibitor. And we treated mice with that and then gave them PD-1, and PD-1 alone, those tumors are completely resistant. They didn't respond. Well, then when you added metformin, they pretty much all cleared their tumors. So I feel like there's a strong metabolic connection in the immune system that is starting to be appreciated.
Well, I'd like to thank you for presenting to us. It's been really great. Should have done 3 sessions.
Yeah, that's all right. We almost got through. I had like two or three more slides.
Okay, and
We're
we
good.
will send those slides
Yes,
out,
we will send
right?
the slides
Okay,
out.
great. All right, thank you
All right.
very much.
Thank you, everyone.